In this particular section, we are going to deal on the cellular mechanisms of innate immunity. We have a separate video on humoral mechanisms of innate immunity, but here we are basically going to discuss only on the cellular mechanisms of innate immunity. So, what is the importance of the cellular mechanism? First is understanding. Second thing, why the cellular mechanisms of innate immunity are important is because you have a separate group of disorders which are linked to this called autoinflammatory syndromes, which are very important for JIPMER exams. Very recently, in the last 3 years, every single paper of JIPMER is having this autoinflammatory syndrome as a very important question. You have a lot of things on autoinflammatory syndromes, even though they are asking very primitive questions. But let us dig in detail, because this particular app is all about your knowledge rather than anything else. That's, I wanted to provide pure knowledge. That's it, as simple as that. So you will get the results if you do it completely. At the same time, you have to understand it very comprehensively also. Very elaborately, many things you have to understand, which is also of paramount importance. That is also what I'm concerned with. So let us see what are the things that contribute to the cellular mechanisms of innate immunity. The first one we already discussed, that's why I'm writing separately, that is called respiratory burst. The first one, respiratory burst, we discussed already. We had a lot of key enzymes like NADPH oxidase. Then we had something called myeloperoxidase, superoxide dismutase. A lot of enzymes we discussed already, which contributed to the respiratory burst, which we already discussed enough in the neutrophil disorders. But that's why I'm not going to discuss about that at all. But second thing, what we have to discuss about the cellular mechanisms is on the HLA and the MHC complexes. HLA, you know, it stands for human leukocyte antigen, and you know MHC stands for major histocompatibility complex. Are both the same? I don't think so. HLA is actually a gene that produces MHC, which is a protein. So which means major histocompatibility complex is a protein that is generally produced by HLA gene. This HLA gene is located on chromosome number 6. It's a multiple time asked question. Everyone knows it's a very primitive thing asked in basic subjects like pathology, microbiology or physiology sometimes. So it's a very simple thing. So chromosome 6 HLA gene gives rise to proteins called MHC. MHC are two types of proteins. You have MHC class 1 proteins and MHC class 2 proteins. What are MHC class 1 proteins? MHC class 1 proteins are generally produced by a group of HLA genes called HLA A, B and C. These are three genes that gives rise to MHC class 1 protein. And MHC class 2 proteins come from a group of HLA genes called HLA-DR, HLA-DQ and HLA-DP. So these are the three genes that give rise to this particular MHC class 2 protein. Simple. So there are a lot of difference between MHC class 1 and MHC class 2 protein. So first let us see in the structure, what are the differences that happen in the structure. So first thing, MHC class 1 has 3 alpha chains. You have an alpha 1, then you have an alpha 2, then finally you have an alpha 3. This is the first difference. And one beta chain in the form of beta 2 microglobulin. But if you take an MHC class 2 protein, you have 2 alpha chains in the form of alpha 1 and alpha 2 and 2 beta chains in the form of beta 1 and beta 2. Remember, they are not structurally similar, these alpha chains and beta chains, but naming wise we are naming it as alpha and beta chains, as simple as that. So this is what is happening in MHC class 1, structural difference, 2 alpha 2 beta in case of MHC class 2 and 3 alpha and 1 beta 2 microglobulin in case of MHC class 1, structural difference. And where is the peptide binding site? So where the peptides are going to bind. So first, the peptide binding site is going to be in the alpha 1 alpha 2 in case of MHC class 1 and in the alpha 1 and beta 1 in the case of MHC class 2. Very important. Peptide binding site in the case of alpha 1 alpha 2 in case of MHC class 1 and alpha 1 and beta 1 in case of MHC class 2. Similarly, you know very well this MHC class 1 is going to interact with CD8 and MHC class 2 is going to interact with CD4 T helper cell. So how will you remember this? Very simple, very primitive, but let me tell you that. So 1:8 is equal to 8 and 2:4 is equal to 8. This is what we call rule of 8, where the product should become 8, where they mention MHCD8 is going to interact with MHC class 1 and your CD4 have to interact with MHC class 2. So this is what we call profit. Very primitive, but just for the sake of telling, I told that. So this is how it interacts. So where the CD4 and CD8 interacts. For example, if you ask me, the CD8 of the CD8 T cell is going to interact with the alpha 3 domain of the MHC class 1. Similarly, the CD4 of the MHC class 2 is going to interact with beta 2 of the MHC class 2. Very important. CD8 of the MHC class 1 is going to interact with alpha 3 of the MHC class 1, and CD4 is going to interact with beta 2 of the MHC class 2. As simple as that. So what are the differences, basic differences between this MHC class 1 and MHC class 2? You can see it here. The basic difference lies in how they present the antigen. You can see the MHC class 1 is actually processing intracellular proteins. Intracellular proteins is what is given in this diagram, but what we are going to discuss is that's an endogenous antigen. That's what is going to be presented with MHC class through MHC class 1. So it's an endogenous antigen. So endogenous antigen means what are the important endogenous antigens that can be presented? Number one is the viral antigen. That is the only way you can present, because virus is intracellular. They have to be presented through MHC class 1 only. And second thing, you have tumor antigens. In tumor, lot of intracellular proteins are going to be aberrant and abnormal. They're not going to synthesize normal proteins. They're going to synthesize a lot of mutated proteins within the cell, and that has to be notified to the police. That has to be notified to the immune system via MHC class 1 only. Again, that is also very important. And the way of processing is also important, because actually these endogenous proteins are actually processed by the proteosomes, not by the lysosomes. Very important. Proteosome, and they are assembled in the endoplasmic reticulum, but entry into the endoplasmic reticulum is given by this protein called TAP protein, which is the one that transfers this processed antigens into the endoplasmic reticulum. Very, very important. TAP is an MHC class 1 pathway protein. And finally, some processing happens in the Golgi apparatus also, and MHC class 1 is expressed, which is actually recognized by CD8 T lymphocytes. The only exception for this is there are certain dendritic cells which process exogenous antigens, and they can express via MHC class 1. Clear? This is to prime the CD8 lymphocytes for extracellular antigens also, because it's not like only intracellular should be presented to MHC class 1. So, and CD8 lymphocyte should be, remembering only intracellular antigens. The dendritic cells also want the CD8 killer cells to be primed for extracellular antigens also. So that is the reason, only exception, a dendritic cell may be, sometimes present exogenous antigens can be presented via MHC class 1. That's the only example. Apart from that, MHC class 1 exams means the endogenous antigen invariably. That's all. And very important second thing is, you can see here, where MHC class 1 is present. If they ask you, MHC class 1 is seen in all nucleated cells of the body. All nucleated cells that contain nucleus. Every single cell that contains nucleus will express MHC class 1. Maybe exceptions the RBC, because they don't contain the nucleus like that. You can do a lot of exceptions, but any single cell that has a nucleus will express this MHC class 1, which might also tell you that MHC class 1 expression is a marker of a self. I have MHC class 1, I am self. That's how the cell tells other people. Between CD8 will not bind with this MHC class 1 if it's a self. Only if it's an antigen is going to bind, because they have been taught not to bind to the self already in the thymus, which we'll discuss in the subsequent sections. So all nucleated cells are going to express MHC class 1. Clear? So there are viruses which try to escape this MHC class 1, obviously, because viruses are very scared of MHC class 1, because viruses will be, exposed, or a cell is going to tell the outside that a virus is there inside me and it's killing me, and they are going to activate the police and the immunity by expression through MHC class 1. So viruses like HIV can down-regulate the MHC class 1. There's a very important defense mechanism of a virus, especially HIV, where you have a protein called NEF protein. This NEF gene product called NEF protein is going to act on the cell, and the function of this NEF gene product is to down-regulate the MHC class 1 protein so that they can escape the CD8 T lymphocytes. Similarly, a lot of tumor cells also can down-regulate the MHC class 1 production. So that they can escape the immunity and they can proliferate. That's the idea. But cells which are down-regulating the MHC class 1, cells which have down-regulated expression or reduced expression of MHC class 1, we have another checkpoint and check process in the form of this will be identified by NK cells. These are called as natural killer cells. Will make sure that the cell which has no or little expression of MHC class 1 should be killed. Why? Because this MHC class 1 is not only recognized by this CD8, they can also be recognized by this NK cells through this special ligands. They have a watch. They see, is this cell really self? I told you MHC class 1 is an expression of a self. So they see that. So any cell that has reduced expression of MHC class 1, they definitely are going to get exposed to NK cells, because NK cells have a watch on this MHC class 1. Once MHC class 1 is not present on a particular cell where it should be present, NK cells identify those cells and kill them. So NK cells are one of the important defense for cells that do not express MHC class 1, especially these virus infected cells and tumor cells which deliberately don't regulate the MHC class 1 complexes. But you might have a question. So how does an RBC escape then? Because a lot of naturally occurring cells in the body also do not have the nucleus. So how these escape which means, they don't have MHC class 1. If they don't have nucleus means like RBCs. So which means they are also exposed to NK cells and NK cells should kill them as well, isn't it? But no, we have another process which we'll be discussing in detail on NK cell mechanism, because it's not only the absence of MHC, it's also an activating ligand that the cell will expose, that will activate the NK cells as well. If they see a cell that doesn't have MHC class 1, it's not only the absence of MHC class 1 that will make the NK cell kill. Anyway, that's a little complicated process we'll discuss later on if possible. But right now understand, any cell that has down-regulated MHC class 1 expression will be exposed to NK cell and subsequent killing will happen. So these are the things that are important for MHC class 1. And next thing, we are coming to the MHC class 2. And MHC class 2 means it's for exogenous antigens. It's for antigens that are present outside the cell, extracellular, exogenous antigens. So what will happen is, here it is not processed via a proteasome or proteasome. Here instead it's processed by an endosome and a lysosome complex. You can see endosome and the lysosome. This is one of the differences that happens with MHC. Here there is no tap protein that is involved typically here like MHC class 1. Here they are processed and they form an endolysosome where the peptides are processed. And from the Golgi apparatus, your MHC class 1 is synthesized, and finally, your MHC class 1 and the peptides unite together. Finally, you can see the MHC class 1 has a bound antigenic peptide here. So this is for the extracellular antigens. And finally, you can see the MHC class 2 is expressed, which is going to be bound by CD4 T helper cell. So this is what is going to happen. So this CD4, once it's activated, once it binds to this MHC class 2 protein, you need a lot of other things also. Co-receptor, co-stimulation is also important. But right now, this CD4, once upon activation, they are going to go berserk and they are going to differentiate into one of the different pathways like TH1, TH2 or TH17 or T regulatory pathway. Depends on what cytokine it is getting from this particular antigen presenting cell. But anyways, and I didn't tell you that fact that this MHC class 2 are typically seen on the antigen presenting cells. At the same time, they are also seen on activated lymphocytes, especially the T cells, activated T lymphocytes. So these are the places. But in exam, typically they will ask you the APs, antigen presenting cells. Professional APs are macrophages and dendritic cells. These are the ones that very typically contain this MHC class 2. Apart from the activated T lymphocytes, also contain MHC class 2. And one thing which you can see here when you discuss in close detail is when they are produced from the Golgi apparatus. These are when they are synthesized. These are called immature MHC class 2 complexes. These are called immature MHC class 2. And when they are producing from the Golgi apparatus, for example, if you have an MHC class 2, and I told you this alpha 1 and beta 1 is the one, this is the antigen binding domain. So what will happen is, when they are trying to produce this MHC class 2 molecule, there is a lot of possibility, because in the Golgi itself you might have some endogenous antigens which has a lot of chance that it might bind with the binding socket of MHC class 2 within the cell. So there's a lot of possibility that endogenous proteins might bind with the socket of MHC class 2. So which means MHC class 2 is for exogenous one, not for endogenous. So this has to be avoided. This is not good actually. Your endogenous peptides attaching with MHC class 2 means there is a lot of possibility the CD4 T helper cell might get activated for endogenous antigen, resulting in autoimmunity, isn't it? So that's not appropriate. So in that case, to avoid that binding of endogenous antigen with the MHC class 2, there is one protective mechanism. That is what this chain is all about. This is what we refer to as something called invariant chain. This is what we refer to as invariant chain. You can see this invariant chain's main principle is to block the antigen presenting site unless and until it reaches the endolysosome complex. Clear? So in the Golgi apparatus there is a protein, this invariant chain also you can call it as clip. That is the one that clips the antigen presenting site, or it blocks the antigen presenting site of the MHC class 2 from binding with endogenous antigens or intracellular antigens. That's the protective mechanism. Later on, certain enzymes like cathepsin L, to be honest. So this enzyme is going to dissolute this, invariant chain, and finally that happens when the endosome and lysosome fuses. The cathepsin L will break down this, and finally your antigen will be binding with the MHC class. So this is an exogenous antigen, not an endogenous antigen. Remember, intracellular antigens are processed by proteasome, and extracellular antigens are processed by endosome and lysosome. These are some of the basic differences. So if they ask you which of the one that contains the phagolysosome, you have to talk about MHC class 2, which involves the proteasome pathway and the TAP pathway. You have to tell about MHC class 1, which is of very, very high importance. And what about the amino acid binding? For example, the peptide that is presented by MHC class 1 has 8 to 10 amino acids. They can bind 8 to 10 amino acids, whereas MHC class 2, the peptide that is presented has almost 13 to 20 amino acid sequences, or 13 to 18 you can write. So 13 to 18 amino acid sequences. So that's what is going to happen. So it's going to be 8 to 10 amino acids for MHC class 1, and it's almost going to be 13 to 18 amino acids, which means the number of amino acids that can be presented, that can bind to the MHC class 1 and class 2 binding pocket are different. Little bit less for MHC class 1, little bit more for MHC class 2. So this is all about, you have to know about the MHC class 1 and MHC class 2. So if they ask you the subtle differences, you have to know which binds with what. That knowledge is very easy, but the most important thing that you might not understand is the proteasome, might forget in the exam rather than not understanding, is the proteasome TAP pathway for MHC class 1 and the invariant chain is for MHC class 2. Number of amino acid residues that bind with MHC class 1, number of amino acid residues that bind with MHC class 2. So these are some of the important things to understand about MHC class 1 and class 2. The next thing what we are going to discuss is on the very important concept called pattern recognition receptors. But the problem with the pattern recognition receptors is that it's really, really tough to understand. So many receptors are there. So our team has created a beautiful diagram on pattern recognition receptors. You have basically 4 different types. This, 4 basically different pattern recognition receptors are there. One is called TLRs, that's called toll-like receptors. Then you have something called CLRs, that's called C-type lectin receptors. Then you call RLR, that's called Rig-like receptors. And finally you have NLRs, that's called Nod-like receptors. These are the 4 basic pattern recognition receptors, which are really, really important for understanding. But the problem is, this is such a beautiful concept, and mind-blowingly they are placed on so much of, strategically they placed inside many parts of the cell. These are typical components of innate immunity, all these pattern recognition receptors, but it's really mind-blowing. So how strategically, how beautifully they are placed in the outside and inside of the cell is something to be really, felt rather than something to be understood. So we can see the toll-like receptors, the CLRs and the RLRs and the NLRs. in that, let us see. So what binds with what? Really, like this is such an interesting topic, because with this also you can understand the autoinflammatory syndromes, which I'm going to discuss, because that's why this is really important. Autoinflammatory syndromes are also associated with problems with a lot of this Toll-like and CLRs, RLRs and NLRs. So let's first start with the TLRs. So you can see TLRs can be presented in two areas, one on the plasma membrane or the cell membrane, and second one, they can be present on the endosome also. You can see they can present on the endosome side also. There are two areas where you can see this Toll-like receptors basically. So what are the Toll-like receptors that are present on the plasma membrane? You can see 1, 2, 4, 5, 6. So all these things, so which means 1, 2, 4, 5 and 6. All these things are going to be present on the cell membrane. In this, the most important things are the 2, 4 and 5. These are the core elements, core things that are present on the plasma membrane, over the plasma membrane. So this is what the TLR, initial TLR is. So what are the things that are present in the endosome? You can see the TLR 7, 8, 3, 9, which means 3, 7, 8 and 9. So these are the TLRs that are present on the endosome. So let us see why these are placed at different places. You can understand very easily if you know what they're going to recognize. So the ligands for this TLR 1 and 2 complex is nothing but the triacyl lipopeptide, and the receptors for TLRs 2 and 6 is nothing but the diacyl lipopeptide. The receptor for the ligand for TLR 5 is the flagellin, and the receptor for ligand for TLR 4 is the lipopolysaccharide mannans, of which the most important regularly asked question is the lipopolysaccharide for TLR 4. Very regularly asked question in exam, even though if others are you don't remember, but this is very important. But rather than understanding what they exactly bind, it's very relevant to understand what these are basically. All this triacyl lipopeptides, diacyl lipopeptides, flagellin, lipopolysaccharide, all are bacterial components. All are coming from the bacteria mostly. That is why they are placed on the plasma membrane. Bacteria extracellular. I'm not telling all. Most of the bacteria are extracellular. And the moment you come into the endosome, you can see what's the ligand for this TLR 7-8 complex. It's single-stranded RNA. What is the ligand for this TLR 9? It's DNA. What is the ligand for TLR 3? It is dsRNA. All these sense viral components. That is the reason why they are present inside the cell most of the time and near the endosome. So you can see very strategically placed, such a strategically placed, receptors. At the same time, majority of the times bacterial complexes are also there, which also can be, processed by the endolysosome, and those DNAs and RNAs can also be, represented by, can bind to TLR that are present on the endosome. So all these things are possible. So very strategically placed, superbly placed. Inside means it's more of a DNA, RNA kind of components. Outside means it's more of a bacterial surface component. Superb, isn't it? So that's how it's placed, so beautifully placed. And similarly, you can see the CLRs. The CLRs, you can see all are placed on these are Mannan-like receptors. You can see this Mannans. This Mannan receptor, the CLRs typically recognize, you can see this beta-1,3 glucan, which is a fungal component, and Mannans again, which are very importantly fungal components. And one of the important receptor type, like how is it TLR 1, 2, 3, 4, like that, you can see there are two important receptors. One is the Mannan receptor, and second is the dectin-1 and dectin-2 receptor, which means you can simply write Mannan receptors and dectin receptors. So there are like TLR 1, 2, 3, 4, 5, 6, 7, 8, 9. You have Mannan receptors and dectin receptors that come under the CLRs, that is C-type, lectin type of receptors. And what they recognize is most commonly, they are going to recognize the fungal components, which means the Mannans, beta-1,3 glucan, all these things are typically the fungal components that these receptors are going to recognize. And come to the Rig-like receptors. Rig1 helicases are always called as RLRs, Rig-like receptors. So what they are going to recognize is the long standard dsRNA and the short strand dsRNA, which could be coming from the bacteria, very importantly from the virus, but it comes from the bacteria also. So short double strand DNA and the long double-stranded DNA. These are what we called as Rig-like receptors. And there are two subtypes of receptors, like how I told you, Mannan and your Dectin receptor. Similarly, Rig-like receptors has two components. One is MDF5 receptor and second one is Rig1 receptor. There are two subtypes of that, like TLR subtypes we thought, and CLR subtypes we saw Mannan and Dectin. Similarly, RLR subtypes are two, that is, you have this MDF5 and Rig1. Finally, you have this NLRs also, this called NOD-like receptors. Remember, NOD receptors are different, NOD-like receptors are different. So here we are talking about NOD-like receptors, these are called as NLRs. Why these NLRs are very important, I'll tell you. You have three types of NLRs, subtypes of NLRs. One is NLRP3, then you have NLRC4, then finally you have something called AIM2 complex. So these are the three different types of NLRs you have. Typically, this NLRP3 is going to recognize this peptidoglycan, DNA, RNA in a candida, and the NLRC4 is going to recognize flagellin, and AIM2 is going to recognize the double-stranded DNA. Leave about all those things. But very importantly, these NLRs form a very, very important pathway inside the cell called as inflammasome pathway. Very, very important. So they form NLRP3 inflammasome, NLRC4 inflammasome, and an AIM2 inflammasome. Like if you want to dig into details, it's too much for you, but I know that, but still that is the reason why I'm going to cut short a lot about this inflammasome pathway. But still, this inflammasome has a lot of importance, because this inflammasome is the one that is really, really important for autoinflammatory syndromes understanding as well. But anyways, you can see that majority of them give rise to something called inflammasome, which is really, really important for the intracellular inflammation, which is affected with tumors or whatever it is. And what they sense, for example, all these receptors we discussed, TLRs, CLRs, RLRs, NLRs, what are they going to sense overall? Even though they have some sense the diacyl lipopeptide, triacyl lipopeptide, flagellin, dsRNA, dsDNA, single-strand RNA, candida, flagellin, Mannans, like beta-1,3-glucan, whatever. What are all this? These are PAMs. What do you mean by PAMs? These are nothing but pathogen-associated molecular patterns. That's what. They ask you what they really sense is nothing but the molecular patterns that are associated with the pathogens. That's why it's called pathogen-associated molecular patterns, PAMs. But they can also sense one more entity called DAMP. That's called damage-associated molecular pattern. They can also sense this DAMP, which is also called as damage-associated molecular pattern. So, what are the DAMPs that can be recognized by the... Till now we have seen the PAMs. Whatever I told you are the PAMs, ligands, that are going to recognize by these receptors, but some DAMPs, internal damage to the cell, that particles also can be recognized by this pattern recognition receptors. What are the DAMPs that are recognized? For example, DAMPs that are recognized include something called HMGB1. HMGB1, this is called high mobility group box 1. So this is a DAMP actually. This is actually recognized by TLR2 and TLR4. And you have uric acid also. This is also one of the important DAMPs that is being recognized by the scientists. So uric acid is recognized actually by this NLRP3. So these are the two important DAMPs that are recognized by the pattern recognition receptors. One is by HMGB1, that is by TLR2 and 4, and NLRP3 recognizes this uric acid. So, but more importantly for exams, these PAMs are the ones that are very, very important. Okay, what they're going to do by recognizing all these things. You can see the final end result of all these receptors, your especially this TLRs, let me put a different color, your TLRs, CLRs and your RLRs, all these three things end up with very, very common thing that is called NF-kappa B. Most of them, you can see them, the end result is going to be increase in this of NF-kappa B. You can see a lot of adapter proteins, a lot of signal transduction molecules in between, but whatever may be the case, the end result is going to be on NF-kappa B. NF-kappa B production will be increased. That is nuclear factor of kappa B, which is a very important thing to induce the production of a lot of pro-inflammatory cytokines. You can see the moment NF-kappa B is activated, you can see a lot of pro-inflammatory cytokines are activated. You can see a lot of pro-inflammatory cytokines will be activated once NF-kappa B is increased in the production. At the same time, there is one more thing that can be increased, that is called this IRF3. IRF3 increase will result in increase in the production of type 1 interferons. One of the important type 1 interferon we saw in the last video may be the interferon alpha. Apart from that, this IRF3 is also increased. There are two things that you have to know, that is NF kappa B is increased, that's the final pathway, and IRF3 may also be increased. Remember, if it's extracellular, it more goes towards NF kappa B related pathway. If it's intracellular, it goes towards NF kappa B as well as IRF pathway. Extracellular cell surface plasma membrane receptors means they more go oriented towards NF kappa B only. If it's intracellular pattern recognition receptors, usually they go towards cytosolic or endosomal receptors, means it goes more towards your IRF3 activation and NF kappa B activation both. But I didn't tell you, in this the toll-like receptors are present on the plasma membrane or the endosome. The C-type lectin receptors are typically located on the plasma membrane, and Reg-like receptors are seen in the cytosol, as well as this NLRs are also located on the cytosol. You can see the diagram and understand easily. It's not a very big thing to understand. So mostly the intracellular cytosolic thing and the endosomal thing, usually they activate both NF kappa B and IRF, but the plasma membrane things usually they activate more goes towards NF kappa B associated pathway only. That's the final thing that's going to happen. But one thing that completely goes in a different direction is the inflammasome pathway, that is the NLR pathway. Usually they don't activate NF kappa B also, they don't activate IRF also, IRF3 also, they're not going to NF kappa B also, they're not going to activate IRF3 also. Instead they are going to activate caspase 1. This caspase 1, finally they are going to activate a cytokine called interleukin 1 beta. Very, very important. This caspase activation followed by interleukin 1 beta activation is what we refer to as something called an inflammasome pathway. So remember, I told you, usually other receptors apart from this NLRs activate mostly NF kappa B or IRF3. But this NLR, this NOD-like receptor inflammasome pathway is going to activate predominantly the caspase pathway and interleukin 1 beta pathway. That's what is going to happen. This is a major difference between these. So you can see now, once this itself looks very complex, but I'll simplify it by breaking into a small table. But this itself might look very complex to you, but let us see in detail about the adapter proteins and all. You can see the most important adapter protein, because I'm not going to discuss all the adapter proteins. One of the important adapter protein is, you can see this MOID88. You can see very, very important MOID88, because in future hematology I'm going to discuss this MOID88 also. It's a very important adapter protein, mutations of which maybe I'll be telling in Waldenstrom's macroglobulinemia and all these other hematopoietic cancers also. But anyway, right now it's one of the very important innate immune pathway system, MOID88, very important adapter protein, MOID88, because mutations of this can result in wide variety of diseases. Very important adapter protein, MOID88. You can see it plays a very vital role in the TLR pathway, the toll-like receptor pathway. If they ask MOID88 is important for toll-like receptor pathway, I'm telling multiple times. Clear? So apart from that, there are a lot of other adapter proteins are there. Again, second maybe important adapter protein that is important is your this pyrin domains. This pyrin domains, you can see this card and pyrin domains, this card and pyrin domains. These are also very important adapter proteins, this pyrin and card. You can see. Why? Because this pyrin gene mutations are associated with the important disease called familial Mediterranean fever, which is a type of an autoinflammatory syndrome. That is why these pyrin domains are again important. Again, two important adapter proteins are pyrin and card. That's it. Especially the pyrin one, that's a very important adapter protein as well. And finally, end result, which you have discussed already. So what in a nutshell I'm going to tell? Let us see in a nutshell. So we can put TLR here, we can put CLR here, we can put RLR here, that is Reg-like receptor, and finally we can put NLR here. So which means TLRs are going to be present on plasma membrane as well as the endosome, and CLRs are typically located on the plasma membrane, and RLRs are located on the cytosol, and NLRs are also located on the cytosol. And what are the things that they're going to recognize? They're going to typically recognize triacyl peptides, which I told you, that is by TLR1 and 2. Then you have diacyl peptides, typically by TLR2 6 complex. Then it's going to be recognized LPS TLR4, and it's going to recognize flagellin, that is TLR5. Then you have double strand RNA by TLR3 and single strand RNA by TLR7 and 8, and finally DNA unmethylated, especially the unmethylated CpG sequences DNA, that is by TLR number 9. So CLRs are going to recognize, very importantly, mannans, beta 1,3 glucans 1,3 beta 1,3 glucans, and Rig-like receptors are going to recognize short strands of double strand RNA, whereas RLRs for long, TLRs are going to recognize long components, whereas RLRs will recognize short double strand RNA and a long double strand RNA as well. They are going to recognize both. And NLRs are going to recognize, as I told you, they are going to recognize flagellin, by NLRC4, and NLRC3 is going to recognize bacterial peptidoglycans, DNA, RNA, Candida. All these things are recognized by, you know very well, these three things are recognized by NLRP3. This is by NLRC4, and aim to recognize is double-stranded DNA, aim to inflammasome. So all these things, these are the ligands for these receptors. But only certain things are important, not all. And finally, you have adapter proteins, the important adapter proteins. Adapter proteins is, here is MYD88 for toll-like receptors, which I have discussed already, because this is the most important as far as I know. For TLRs, it's a very important adapter protein. Apart from that, you have TRIF, TRAM, all these things are there. Remember, almost all the TLRs, this is also very potential MCQ, all the TLRs use this adapter, that is MYD88 adapter, except TLR3. Only TLR3 will not use MYD88 as adapter. Remaining every single TLR will use MYD88. You can see here. You can come back. You can see TLR3. They use only TRIF as an adapter. They don't use MYD88 as an adapter, whereas all the other TLRs are using MYD88 adapter. Very, very important. This is a very important adapter protein. And next thing is for what is the adapter for CLRs? It is SYK. It's a tyrosine kinase. Then what is the adapter for RLRs? RLR adapter will be your IPS1 and FADD, not again important. And here the important adapter is ASC, that contains the card domain. So this is the most important adapter, ASC and the card domain. So which is called as very important, because ASC is the one that's going to bind with the pyrin, deficiency of pyrin, mutation of, not deficiency, mutation of pyrin is what we call it as the familial Mediterranean fever. And there are a lot of signal transduction proteins. What are the proteins that help in signal transduction? Signal transduction proteins are nothing but, you have IRAK1, you have IRAK4, and the signal transduction proteins here are CARD9, and the signal transduction proteins here are TRAP3. And here you don't have that much signal transduction proteins, but more importantly, I'll tell you the inflammasome pathway separately. So once the signal transduction proteins are activated, what you are going to increase? You are going to increase the production of NF-kappa B, plus or minus you are going to increase the production of IRF3. Out of which the NF-kappa B is the most important. That is where everything terminates. What NF-kappa B is going to do? What is the reason for increasing NF-kappa B? Because it's going to increase the transcription of certain mRNAs, which will translate into proteins later on. What proteins will be increased in the cell once NF-kappa B increases? These are the pro-inflammatory cytokines like interleukin 1, interleukin 18, then you have tumor necrosis factor alpha, interleukin 6, MCP1. All these things are very important pro-inflammatory cytokines. And IRF3, they are going to increase type 1 interferon production, which we already discussed. Type 1 interferon, especially interferon alpha production, is increased with the help of IRF3. So what CARD9 is going to do? This CARD9, TRAP3, once again, they are going to increase the NF-kappa B production only. This also we discussed already. Everything is going to terminate with NF-kappa B. But NF-kappa B is not related to the inflammasome pathway, that is NLRs. So what they are going to do is, so I'll tell you first, ASC will be activated, especially the card domain of ASC will be activated. Once ASC is activated, they will activate the caspase 1, caspase 1 activation. Once caspase 1 activation happens, they will increase the production of IL-1 beta. How they do that is by cleaving this pro IL-1 beta. The pro component of the pro IL-1 beta will be removed and they will form active IL-1 beta. And second thing, they also produce the same thing is they can increase something called gasdermin D. Like IL-1 beta, the pro-gasdermin D, there will be a molecule inside the cell. That pro component will be cleaved by the caspase and you will produce gasdermin D. Gasdermin D will cause pores if the cell is very badly damaged by the virus or some other damage, because that cell should die. So this gasdermin D will introduce pores within the plasma membrane and water influx will come, and that also is a pathway for releasing a lot of these inflammatory cytokines that are already building up inside the cell, and the cell typically dies. So that's what gasdermin D does, inserting pores into the plasma membrane. So all these things are going to happen. So these are the, like in a nutshell, you can tell about all this TL pattern recognition receptors, TLR, CLR, RLR and NLR. So I did it's a pretty tough thing to understand, maybe a little dry also, but still really important. It's such a pattern to understand all these pattern recognition receptors. This is how you understand. These are very beautifully placed in overall the cell. And there are other receptors which I have not mentioned, which I'll tell you in just a minute. What are the other receptors? Number one, you have something called RAGE. This is for receptor for advanced glycosylation end products, which is actually one of the receptors are called DAMP receptor. It's not a PAMP receptor, it's actually a DAMP receptor, and it's seen on the plasma membrane. Again, it's a plasma membrane receptor like toll-like receptors and CLRs, C-type lectin receptor. This is also plasma membrane receptors which typically recognizes the advanced glycosylated end products in diabetes. That's called RAGE receptor. This is a DAMP receptor actually. And second thing, there's something called NOD1 and NOD2 receptor. Remember, these are not NLRs. These are not NOD-like receptors. NOD-like means not like, not NOD. NOD1 and NOD2 are separately different. These are typically intracellular receptors, intracellular and cytosolic receptors only. They are not extracellular receptors. What NODs they do is, NODs recognize certain substances, like for example, ligand for NOD1 is peptidoglycan. Peptidoglycan, very important for your Crohn's disease and Blau syndrome understanding, especially Crohn's disease. If you take any book and see this NOD1 mutation, they would have given many cases. So that's why NOD2 mutation in Crohn's disease. You can see any textbook you can take and see these NOD mutations will be definitely given in Crohn's syndrome. You can go and see. So now you can see NOD2. What about NOD2? NOD2 recognizes something called muramyl dipeptide, which is also a bacterial component. Muramyl dipeptide. Here the adapter will be something called RIP2, RIP2, that is adapter here, like how MODD, we discussed about MODD, then we discussed a lot of other adapters also. Here like that, RIP2 is a very important adapter. Once it's activated, it will increase the production of, again, the same old friend, that is NF-kappa B has come again. This NF-kappa B is going to increase the production of pro-inflammatory cytokines, which is going to have a lot of effects. And the very, very important thing to understand is this NOD1 and NOD2 is not going to act via inflammasome pathway, which means they are not going to activate caspases, they are not going to follow that IL-1 beta pathway separately. They are going to follow like other pattern recognition receptors like TLRs and CLRs, where they are going to activate only the NF-kappa B. Remember, inflammasome NLR, not like receptor pathway, do not follow the NF-kappa B pathway, they are caspase pathway. But NOD receptors are actually standard PRRs, they are not going to follow the inflammasome and NF-kappa B pathway, which is really, really important to understand. Remember, this NOD2, especially the CARD15 domain, NOD2 CARD15 domain, you can see here in the NOD2 you have this, NOD2 is not mentioned, sorry for that. So sorry for misdirecting you. NOD2 is not mentioned here inside the cell. We have not mentioned that in the diagram. So NOD2, especially the CARD15 domain of NOD2, may get mutated, and this is the reason for developing something called Crohn's disease. And if you want to know another autoinflammatory syndrome that gets developed, that's called Blau syndrome. That is also due to this NOD2 CARD15 gene mutation. That is why this NOD2 and CARD15 gene is also called as IBD1 gene. IBD1 gene. This CARD15 gene and IBD1 gene is present on chromosome number 16, which I will again tell you in the GIT section about Crohn's disease. This is NOD2 CARD15 mutation, which is very important for developing Crohn's disease later in life. That is present on chromosome number 16. So once we have understood all these things, let us see the diseases that are caused due to PRRs. First, let us see this is due to PRR defect and deficiency. For example, if PRR is defective or deficiency, so certain PRR is defective or deficient, so what will be the diseases that you will get? I mean, disease or what could be the infection susceptibility. For example, there are long term, there was no clue for understanding certain things. For example, suddenly a patient who's coming to the OPD, you might be seeing a terrible bronchiectasis without risk factors, and there will be Pseudomonas colonization. And suddenly one patient will be coming with Listeria monocytogenes. All of a sudden, no risk factors. You don't know why. You just scratching the head. You study in the books, risk factors for Listeria is something, risk factor for Pseudomonas is this thing. But a patient who comes without any risk factor and gets infected with all these organisms, what could the underlying reason could be a PRR defect. That is why they might get an infection even without a risk factor. You see a lot of cases in the OPD, you don't do the mutation testing, that's why you don't realize that this is the fact. And these patients do well for most of the time unless until the defect is very severe. So we even don't care much about it, treat the patient and send them home. But there could be an underlying PRR defect or PRR mutation which we have no idea and we are not going to test it for as well. So that is why it is important to understand this. What could be the underlying PRR defect which might increase susceptibility to a particular infection? Why a patient goes for particular infection susceptibility, that's what we're going to discuss. First one, what will be the MID88 defect? Remember, I told you MID88 is important for all the TLRs, it's an import adapter except for TLR3. So which means it's not going to affect the TLR3 only, but it's going to affect all the TLRs, which means infection susceptibility is typically for pyogenic bacteria. Any pyogenic bacteria, your infection is going to happen. So what are the characteristic pyogenic bacteria, pus forming bacteria? Pseudomonas, pneumococcus, staphylococcus, streptococcus. So these are the typical pus forming bacteria as far as I know. So these are the important things that they are susceptible for. So probably you can put bar IRAK4 deficiency also, because IRAK4 mutations, IRAK4 is associated, it's a signal transducer protein that is associated with MOD88. So you can remember as MOD88 IRAK or simply MOD88 deficiency or defect. So second one, what will be the TLR3 defect? TLR3, remember, they recognize the double stranded RNA. So they are going to increase the susceptibility for viruses because double strand RNA. So that is why increased risk for herpes simplex virus, encephalitis. And third one, TLR5 defect. TLR5 is for recognizing flagellin, which discussed already. Listeria is the one that's very important, which has a typical motility due to flagella only. So Listeria infections are very high in patients who's having TLR5 defect. And number 4, you have something called Dectin1. Now you are moving into the CLRs. Dectin1 defect. Dectin1 recognizes beta D glucan. Everyone knows that. So what will be the result? They are going to have increases of Candida and trichophytons. Typically Candida. Candida is the most important. Candida. And number 5, you have this CARD9 mutations. CARD9 is going to recognize this beta glucan again. So once, because you can see, like CARD9 is here in the CLRs only. You can see this. Yeah, CARD9, it's in the CLRs. You can see this. So CARD9 defects are typically CLR defects, and again they are going to recognize this beta glucan only, and again you are going to have the same old story. Candida infections are very high. And you have MBL, mannose binding lectin. It's a complement activation defect where you'll have infection to bacteria and fungi as well. Then number 7, you have NOD2 defect. I'm not talking NOD2 mutation, we are talking about NOD2 defect. NOD2 typically recognizes the peptidoglycan, I told you, recognition defect, and typically you see in Crohn's disease. Peptidoglycan and muramyl dipeptide as well. So peptidoglycan is recognized in NOD2 also, Crohn's disease. NOD2 defect, this I told you, it's a local defense defect in the gut that leads to Crohn's disease. Number 8, NLRP3 defect. NLRP3 is very important for producing the IL-1 beta, caspase pathway. So IL-1 beta will not be produced, so that you will result in autoinflammatory syndromes. These are called as autoinflammatory syndromes. In this, everything is autosomal dominant. All are autosomal dominant. For example, MOD88 is autosomal dominant, TLR3 is autosomal dominant, and NLRP3 defect is also autosomal dominant. But this TLR5 defect, this Dectin1 defect and MBL defect, all these things are autosomal recessive defect. Clear not? So these are the most important: autosomal dominant and autosomal recessive defect. Not two defects maybe inherited in different pathways we'll discuss, but these are the autosomal dominant and autosomal recessive defect. These are the PRR defect and infections that you are susceptible for. And final thing that you have to know is on, we are moving to the, we are going to nail this one. This is what we refer to as something called autoinflammatory syndromes. Autoinflammatory syndromes. Very importantly, the first thing you have to understand because it might sound to you like it's an autoimmune disorder, autoimmune syndrome. It's not, because autoinflammatory syndrome is different from a autoimmune disease by very simple thing, because the autoinflammatory syndrome is due to problems of the innate immunity, whereas autoimmune syndromes or autoimmune diseases are due to problems of the adaptive immunity. Very simple. That's why it's very important, because even though it's rare, autoinflammatory syndromes compared to autoimmunity, but still this is the basic difference. Autoinflammatory are due to problems of the innate immunity, and autoimmune are due to problems of the adaptive immunity. And your MHC association is usually seen with autoimmune, isn't it? There is no MHC association. You cannot tell even a single MHC association, because MHC is not related to your innate immunity, because it's only a bridge between innate and adaptive immunity, but it's not actually typically a innate immunity in the first place. It's only a bridge for the adaptive immunity. So MHC association is typical of autoimmune diseases, but MHC association is not seen in case of autoinflammatory syndromes. So what are the autoinflammatory syndromes that you have to know? So whenever the moment I talk about this autoinflammatory syndromes, let me tell you the basics. You're going to have three important picture. So number one, lot of patients will be having skin and mucocutaneous manifestations. Definite skin and mucocutaneous manifestation will be there, almost every single patient. Second, some musculoskeletal. When I write MSK, it means it's musculoskeletal. Musculoskeletal manifestation surely will be there, for sure. Plus or minus they might have fever, and plus or minus they might have other manifestations also, which we'll discuss. This is how I'm going to categorize the features of the autoinflammatory syndrome, where skin and mucocutaneous with musculoskeletal involvement is almost typical of all the autoinflammatory syndromes. Fever, almost every syndrome will have fever, with a few exceptions are there, and a lot of other features are also there among these syndromes. So these are the basics of the autoinflammatory syndrome. Whenever you think about an autoinflammatory syndrome, you can divide into basically 3 to 4 types. One is the periodic and recurrent fever syndromes. Periodic and recurrent fever syndromes. Second one, I'm going to put under something called cryopyrinopathies. Cryopyrinopathies. And third one, you have pyogenic disorders. And fourth one, you can put under miscellaneous disorders. Fourth one, I can call it as miscellaneous disorders. So what are the very important things to understand? So remember, whatever I told you, this all four, this skin, musculoskeletal, fever, others, everything, all this will be there in periodic recurrent fever syndrome, which means they will have skin manifestation, they will have musculoskeletal manifestation, they will be having fever, and they will be having other organ manifestations also. But if you ask me cryopyrinopathies, in cryopyrinopathies, two things are very important. Almost all cryopyrinopathies are due to your inflammasome, NLRP3 mutations. Very, very important. All are due to NLRP3 mutations, every single cryopyrinopathy which I am going to tell. And second thing, every single cryopyrinopathy will have urticaria as a major manifestation. So skin manifestation, that is predominant. They will have a lot of other things also, plus other things also will be there, others also will be there. But every single damn thing in cryopyrinopathy will have urticaria. And skin urticarial rash will be there. And the characteristic feature of this pyogenic disorders is that they do not have a fever. This is very, very important. They don't have fever. No fever is characteristic of this pyogenic autoinflammatory syndromes. Besides, you have a lot of others which will, like this variable picture will be there, which we'll discuss in some time. So no fever. So these are things. So everything will be there in periodic recurrent fever. Urticaria is typical of cryopyeloneuropathy and no fever in pyogenic. That's why I told you again and again, even though they are called pyogenic disorders and they have a lot of pustule formation, they do not have fever typically. Very important point. No fever. If they ask you in exam, all of the following has fever, all of the autoinflammatory syndromes will have fever, except answer must be pyogenic disorder, which I will tell in some, because under them itself you have a lot of things. For example, you have a lot of periodic recurrent fever syndromes. If you ask me what are the periodic recurrent fever syndromes that are important for knowing them, this is called TRAPS. First one is called TRAPS. Second one is the familial Mediterranean fever. TRAPS means it's a TNF receptor. So TR stands for TNF receptor associated periodic fever syndrome. That's what we call TRAPS. That's called TNF receptor associated. It's a gain of function mutation, or TNF receptors cannot be degraded because of the mutation in the TNF receptor. So the TNF receptor will be excessively stimulated by TNF alpha. Very simple. So that's the reason. So TNF alpha receptor associated periodic fever syndrome is what we call TRAPS. And you have FMF, that is nothing but familial Mediterranean fever. And third one, you have something called HIDS. That's called hyper IgD syndrome. It's called, I'll put hyper IgD syndrome or hyperimmunoglobulin syndrome. That's called HIDS. And fourth one, you have something called a NOMID. NOMID will not come under here. So fourth one, you have something called FAPA syndrome, PFAPA. This is a thing that stands for a lot of things. You have periodic fever, then aphthous stomatitis, pharyngitis, and adenopathy. Periodic fever, aphthous stomatitis, pharyngitis, and adenopathy. Adenopathy usually cervical lymphadenopathy. That's what we mean. This is called FAPA syndrome. The FAPA syndrome, other name for that is also called as Marshall syndrome. And fifth one, you have something called AOSD. This is a very simple thing, adult onset Still's disease, which is an equivalent of childhood terminology called like Still's disease in childhood, otherwise called as SOGIA. That's called systemic onset juvenile inflammatory arthropathy. That is otherwise called as Still's disease. So there are two equivalents are there. One is called adult onset Still's disease, which happens in adults more than 16 years of age. If the same disease spectrum happens in children, that's called simply Still's disease, or you can call it as SOGIA or systemic onset juvenile idiopathic arthritis. So this is what we call, these are the different periodic fever syndromes that are associated. And coming back to the cryopyeloneuropathies, if you ask me what are the cryopyeloneuropathies, you have to know. There are three things that are very important. One is called FCAS, that is familial cold autoinflammatory syndrome. That's all. It's called familial cold autoinflammatory syndrome. And second one, you have something called Muckle-Wells syndrome. Muckle-Wells, MWS. That's called Muckle-Wells. If you want, I will write that is Muckle-Wells syndrome. And third one, you have something called NOMID or SINCA. NOMID means neonatal onset multi-inflammatory disease, or neonatal onset multiple system inflammatory disease, or multiple systems will be involved. Or SINCA stands for chronic infantile neurologic cutaneous and articular syndrome. But I don't think in exam they will ask you to expand most of the times, but I feel still if they ask you to expand, it must be JIPMER exam most of the time. That's where they ask these kind of questions. But apart from that, this NOMID and SINCA, they won't ask you to expand. Instead, they will give the syndrome itself. Usually the term preferred in exam will be the NOMID, neonatal onset multiple system or multi-inflammatory disease. And you have the pyogenic disorders. Pyogenic disorders are again three pyogenic disorders are there. Number one, you have something called PAPA syndrome, that is called pyogenic arthritis and pyoderma gangrenosum and acne. So pyogenic arthritis, pyoderma gangrenosum and acne, this is what we call PAPA syndrome. And second one, you have something called Majeed syndrome. And third one, you have something called CRMO, that is called chronic recurrent multifocal osteomyelitis. It's a bone problem. Chronic recurrent multifocal osteomyelitis. This is what we call CRMO, which comes under pyogenic disorders. Miscellaneous are there. Miscellaneous, you have something called some granulomatous autoinflammatory syndromes are there. One of the best examples of this granulomatous autoinflammatory syndromes is Crohn's disease. Simple. And second one, you have something called a Blau syndrome. I mean, majority of the features of the Crohn's and Blau syndrome will be similar, but one small exception will be there. One small exception means Blau will not have intestinal features, which is very typical of Crohn's. Crohn's can be diagnosed based only on intestinal features, isn't it? So both are due to that NOD mutations, NOD2 mutations, CARD15 mutations. But Crohn's is typically, like having intestinal. That's what is Crohn's is all about. But Blau is having almost every single feature of Crohn's, but there will be no intestinal features. That's what about Blau. Both are autoinflammatory syndromes. Both are due to NOD2, CARD15 mutations. At the same time, Blau, it's inherited, right? Blau is inherited, typically due to autosomal dominant, whereas this Crohn's is not inherited. Crohn's is not an inherited disease. Very, very important point. Crohn's is never inherited disease. Most times you don't see the family history in Crohn's at all. Simple, isn't it? So it's not inherited. Even the genetic elements are there, but it's not inherited. And next you have something called DIRA syndrome, that is due to deficiency of something called deficiency of IL-1 receptor alpha, so otherwise called as IL-1RA. So this is what we call DIRA syndrome. And third one, you have something called DITRA syndrome. So this is nothing but deficiency of IL-36 receptor, not alpha, antagonist. IL-36 receptor antagonist. And remember, these are separate disorders, but these are pustular disorders. These are pustular disorders. Rather than, I told you these are granulomatous syndromes, similarly these two are pustular syndromes. We'll come back to that. So this is what we call DIRA and DITRA. DIRA and DITRA can be put under pustular syndromes, but still I won't tell it on miscellaneous because these are only the names will be asked. DITRA means, why they call DITRA means this is called deficiency of interleukin 36 receptor antagonist. For 36 only they have put this T there. That's a deficiency of interleukin 36 receptor antagonist. So these are the various classification of autoinflammatory syndromes. You have periodic recurrent fever syndromes, you have cryopyrinopathies, you have pyogenic disorder syndromes, and you have miscellaneous syndromes. So these are the very basics you have to understand. I will take another 8 to 10 minutes of yours to discuss on certain important manifestations of these disorders. For example, this itself will make a lot of sense. This itself will give you a good understanding about how to approach the autoinflammatory syndromes if they ask in exams. 99% of the times with this itself you can answer all the questions. But to be completely as a knowledge pack, I'll tell you some details about all this other syndromes also. Like for example, if you have a TRAPS, typically the fever, when the fever comes, I told you all these things will have fever, isn't it? All these periodic fever syndromes will have, that's why they are called as periodic fever syndromes, they'll have fever. So fever is not seen only in pyogenic disorders, pyogenic syndromes, but here definitely fever will be there. So what will be the fever? Fever typically happens once in 1 to 3 weeks. Fever lasts for 1 to 3 weeks. Here fever lasts for 1 to 3 days, not 1 to 3 weeks. In heats it is 3 to 7 days of fever will be there. And in FAPA there will be 4 to 5 days of fever, and there will be a typical cycle will be there every 4 weeks. Every month they will get fever, which will last for 4 to 5 days approximately. And very importantly, this is the only syndrome, this SOGIA or AOST, they will have daily fever. This is also referred to as quotidian fever. This is a daily fever syndrome. This is what we refer to as AOST. Very, very important. Very, 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 very important. Daily fever. Daily fever. Very important. That is what happens. Quotidian fever. Other things, they will have a cycle. They will have some 1 to 3 weeks of fever, then they will become afebrile. Here 1 to 3 days of fever, then they will become afebrile. 3 to 7 days of fever, then they will become afebrile. Here 4 to 5 days, then again they will be afebrile for the next 1 month. All have a cycle. But AOSD doesn't have a cycle. Daily fever will be there, followed by afebrile period. They don't have any, days together of afebrile period. Every day they'll have fever. So this is what about SOGIA is. And where will you get this? What is the mutation that is associated with this TRAPS? If you ask me in TRAPS, what is the mutation? It is TNF receptor alpha, or I can simply write as TNF receptor 1 gene mutation, autosomal dominant in inheritance. Familial Mediterranean fever, it's due to MEF-V gene mutation. Again, it is autosomal recessive in inheritance. Remember, this MEF-V gene gives us a product called pyrin. I told you it's a very important component of inflammasome. That NLRP4 and NLRP3, isn't it? So very important component of especially NLRP3 inflammasome, pyrin, very, very important, isn't it? Pyrin. Pyrin mutation. So that's what happens in familial Mediterranean fever. And in HIDS, it is due to MVK gene, that is called mevalonate kinase gene, which is cholesterol metabolism problem. There it will happen. And again, it is autosomal recessive in inheritance. And in FAPA, we don't know what is the reason. We don't know what is the reason for the genetic association, and it is an acquired disease. Remember, it is not an inherited disease. Adult onset still's disease is an acquired disease. If they ask you which of the following is acquired, your answer must be adult onset still's disease or SOGIA or simply still's disease. All these are acquired, this is not inherited. The first three are typically inherited. This could be the question. And I do expect this pyrin gene. This is very important for exams because my instinct is telling, my gut feeling is telling that pyrin might be a very important thing for exams because this disease itself is very important favorite for examiners most of the times. This familial Mediterranean fever. And onset also varies. A lot of things are there, but only certain other features I want to tell. All these things will have musculoskeletal features, skin rash, aphthous ulcers may be there, palmar plantar rash will be there. I'm not going to talk about. See, fever I told you will be there, and I told you the genetic associations. All this will have skin, arthritis, musculoskeletal mutations. I'm not going to discuss about any of those things. Commonly, you can remember they will have definitely skin rash, maybe erythematous, erysipelas like aphthous ulcers, any skin rash you might encounter with this. Arthritis, definitely you will get myalgia, arthralgia, arthritis, erosive, tenosynovitis, any musculoskeletal feature. I'm not going to talk about all those things. I told you already in the common thing, skin, musculoskeletal will be there, fever will be there, but other manifestations, what will be the most important other manifestation? That's what is really important for this. If you ask me, in familial Mediterranean fever, the single most other manifestation is they are going to come with serositis, chronic serositis. They're going to have recurrent serositis. That is typically they'll come with abdominal pain, and they can have a scrotal swelling. Abdominal pain, they'll have scrotal swelling. Remember, I'm not going to talk about skin rash. It will be there in FMF. They're going to have arthritis. I'm not going to discuss about that, but this others, serositis and scrotal swelling are very important. And these FMF patients are very, very at high risk of getting amyloidosis. Very, very important point. Among all the autoinflammatory syndrome, the one autoinflammatory syndrome which has the maximum chance of going for amyloidosis, I'll go for FMF, familial Mediterranean fever. One of the important reasons for death is amyloidosis and cardiac amyloid deposition and arrhythmias and death. That is the reason for death in FMF most of the times. I've seen so many cases, presentations in CMEs where practitioners present this FMF with an ECG, starting with an ECG, because they wanted to show the patient has amyloidosis. Amyloidosis in ECG will show restrictive cardiomyopathy. Restrictive cardiomyopathy in ECG will show low voltage complexes. If they wanted to start FMF as a case, they will show an ECG having low voltage complexes, which is suggestive of restrictive cardiomyopathy because it is due to amyloidosis. That is the reason for death in FMF. So that is the reason why I'm telling you the amyloidosis is so important for familial Mediterranean fever. So then you have something called TRAPS. In TRAPS, I don't think much is important, and again, it's something other thing that is important, the serositis, abdominal pain, this periorbital edema, conjunctivitis. But anyway, serositis is again a feature of TRAPS. And how will you differentiate between FMF and TRAPS in general? Because if both are going to come with serositis, skin manifestations, abdominal pain, serositis and all, how will you differentiate? Simple. Remember, amyloidosis will not happen, but that's a long shot. Over a long time only you will get amyloidosis in FMF. But yearly, how will you diagnose? Very simple. TRAPS typically happens in one particular age group. TRAPS happen in only, I'll put a different color. Maybe TRAPS happen in usually infants. That's the key. Infants. They might look like FMF, but this happens in infant. And FMF typically happens in teenage and young adults, maybe 20s, 15s, 10s. So this is the age, teenagers and young adults. This is where you get FMF predominantly. But infants go for TRAPS, similar to FMF. HIDS, one of the important things about HIDS, they will give a clue in exam is on, maybe they'll give a very important clue in exam. They may tell you that the patient is having elevated IgD, as well as patient will be having elevated IgA. And very importantly, patient might be having cervical lymphadenopathy. Cervical adenitis, this is the key. Cervical adenitis, remember the patient is having fever with cervical adenopathy, And heats usually present typically in less than 2 years of age. Within 2 years of age they present, very young age again. But the key will be on elevated IgD and IgA levels along with cervical lymphadenopathy. That is what you call about heats as a very important thing. And this FAPA also may be presenting very similar, because in FAPA also what you will see is very importantly you will have cervical adenitis. Cervical adenitis will be the key, cervical adenitis, and prominently they will give aphthous ulceration. Aphthous ulceration, because expand FAPA and what you will get. What is FAPA? You have periodic fever, aphthous stomatitis, pharyngitis and adenopathy. So that is the reason why the cervical adenitis and aphthous will be very prominent in case of FAPA syndrome. But if you really wanted to know one important thing about this FAPA syndrome, it is one of the easily treatable through a very simple process called tonsillectomy. And there are a lot of cases tonsillectomy, showing that FAPA syndrome is 100% responsive to tonsillectomy. These are all, see, when you want to discuss about the autoinflammatory syndrome, I can discuss for hours together, but these are the important points. Tonsillectomy cures FAPA. Simple. FAPA has no genetic underlying known thing. Till now, we don't know anything about genetics. Expand FAPA, periodic fever. Then you have, typically if tonsillectomy means which age group? Children will be affected in FAPA. And they'll have aphthous stomatitis, then they have pharyngitis and cervical adenopathy. Simple, that's it. This is FAPA, and tonsillectomy cures them. Most of single dose steroid. All these things are very easy to treat, that's why. And adult tonsillitis disease is a separate disorder, which I have also discussed in rheumatology, where adult tonsillitis disease means, can happen in juvenile age group also, but adult tonsillitis disease means it should happen at age more than 16 years, number one. But JA can happen in age less than 16 also. We are talking about only about AOSD, not that SOGIA. Age more than 16, patient will have a daily daily fever. By definition, they should have at least for fever for more than 1 week, they will have daily fever. At the same time, they will have rash. This typical rash in Still's disease, that is adult onset Still's disease, we call them as salmon rash. It's a pink evanescent rash, salmon colored rash, very, very classical history in exam, salmon rash. And they'll have arthritis for sure. Obviously these are all features of this periodic fever syndrome. I told you everything will be there. Arthritis is a very classic key, and they will have extra other features as lymphadenopathy and hepatosplenomegaly. And one of the very important features of this AOSD is they will have extremely high ferritin levels. This is not a criteria, but still very high ferritin levels is one of the keys for diagnosis. If they give arthritis with ferritin levels of 10,000, why the examiner wants to give that ferritin levels is because he wants you to diagnose AOSD there. Adult onset. And many times one important thing that many books will not give you is AOSD comes as PO in adults, pyrexia of unknown origin. In fact, all the periodic fever syndromes will have PO only. They will be presenting like PO only. But this adult onset Still disease is one of the characteristic adult disease that comes like PO, pyrexia of unknown origin. Very important, AOSD will be happening after 16 years of age, but it will be within 35 years. This is also not given by many textbooks, which means you cannot diagnose AOSD after 35 years of age. It's very difficult to diagnose. So 16-35 is characteristic, and this daily fever, salmon rash, arthritis, then you have lymphadenopathy and hepatosplenomegaly with extremely high ferritin, very, very high ferritin. Diagnosis based on a criteria called Yamaguchi criteria. We have discussed in detail on rheumatology section. You have something called a Yamaguchi criteria, that is the criteria given for diagnosing adult onset Still's disease, not Still's disease, not SOGIA. SOGIA is just Still's disease. And what are the important facts? These are the important facts, even though this looks very messy right now, but you know very well. So the key points I want to take is TRAPS in infants and familial Mediterranean fever in adults. Young adults, maybe in the 15s or 20s. And amyloidosis means familial Mediterranean fever, most common cause of death, high risk of amyloidosis in familial Mediterranean fever, MEF gene pyrin deficiency, and FAPA syndrome, the cervical lymphadenopathy, tonsillectomy cures them, and single shot steroid might also cure FAPA syndrome. And finally, adults on Still disease diagnosed by Yamaguchi criteria, very high ferritin and all the characteristic features I told you, the salmon rash and all these things. And you have HIDA or HITS, where you have elevated IgD and IgA as a key with recurrent fever. At the same time, very small children, less than 2 years of age in HITS with cervical lymphadenopathy. So this is how you diagnose it. It's very simple. All these other features I told you. But if you want this, you remember. But apart from that, I feel it really important. So that's why I'm telling you all this. I've just scratched the surface of the autoinflammatory syndromes, but I'm telling only the important points of how we make a diagnosis in our routine day-to-day practice. I've done a lot of case discussions on autoinflammatory syndromes till now because it's increasingly recognized. Even in India, we have, even in my case, I have described like 3 to 4 cases we have described in a lot of meets, and even I have put some 1 or 2 cases in JAPI also. So it's becoming really common nowadays, autoinflammatory syndrome. That's why the questions are also coming on that. So FCAS, this is familial cold urticaria, and Muckle-Wells syndrome and NOMID. So let us see what are the important things that you have to know about all these 3 things. So first thing, remember this Muckle-Wells and this NOMID will have one important all these 3 things will have urticaria. I'm not talking about the urticaria with autoinflammatory, with recurrent fever, go for some familial, this cryopyrinopathy is not autoinflammatory syndrome, but what will be the clear-cut feature of this Muckle-Wells and NOMID is they have sensorineural hearing loss and they have uveitis. This is very, very, very important. This is what the key for diagnosing the sensorineural hearing loss and uveitis. And this FCAS will not have sensorineural hearing loss and will not have uveitis. No uveitis, and they may have conjunctivitis, like because the form of urticaria allergy, and you might have not said conjunctivitis, it's allergic, but no uveitis, no sensorineural hearing loss. Very important. First you split like that. Then how will you separate these 2? This Muckle-Wells and NOMID. See, remember, typically, this NOMID is going to happen within 1 year of age. This FCAS is also going to happen in infants less than 1 year of age. But Muckle-Wells typically happens in infants more than 1 year of age. In children or adults more than 1 year of age. It's not going to happen in infants, definitely. So that's one way of separating out this Muckle-Wells and NOMID. Second thing that takes NOMID and makes it stand out is the CNS involvement. CNS, typical of NOMID. CNS involvement. They might range from headaches, papilledema, mental retardation, then optic atrophy due to chronic papilledema, obviously. Then mental retardation, all these things. The CNS manifestation is the key. That is the one that makes NOMID and CNCAS stand out from all these other 3 cryopyrinopathies, because CNS involvement is so classical of NOMID, happens in infants. That's why it's called neonatal onset multi-inflammatory disease or multiple system inflammatory disease, less than 1 year's CNS manifestation with sensorineural hearing loss and uveitis. Along with it, they will have urticaria as well. Whereas FCAS will not have sensorineural hearing loss and no uveitis, only urticaria, that itself will clearly grow. They might have some musculoskeletal manifestation, but 1 year old child, how will you tell musculoskeletal? Very difficult. Muckle-Wells, older age, have sensorineural hearing loss and uveitis, but they have this arthritis, myalgia and all, but they do not have this characteristic CNS involvement apart from this sensorineural hearing loss. No CNS involvement. That's how you separate Muckle-Wells out of the equation. And Papa syndrome, if you take, so how can you tell it's Papa syndrome? Papa syndrome name itself tells that it is pyogenic arthritis, then you have pyoderma gangrenosum and you have acne. So these are the features. That itself we can tell pyogenic arthritis because they're pustular, so that's why it's pyogenic. And acne is also pustular condition. And in between you have pyoderma gangrenosum. All these 3 things are pustular condition. That's why it's called Papa syndrome. If they ask you the gene that is responsible for Papa syndrome, that is something called PST-PIP1 gene. PST-PIP1 gene, which is inherited as autosomal dominant in inheritance. I already told you all the cryopyrinopathies are due to NLRP3, but I didn't tell you the inheritance. These are autosomal dominant. Very simple. All are autosomal dominant. And this is, except this NOMID. NOMID is supposed to be, one more difference is it's sporadic. It's not inherited. It's a sporadic disease. I didn't tell you that. But in general, you can remember all are due to NLRP3 mutations. NLRP3 mutations you get in cryopyrinopathies. But Papa is due to PST-PIP1 and autosomal dominant inheritance. And Majeed syndrome is due to LP1N2, and it is due to autosomal recessive inheritance, LP1N2 gene. And CRMO, we don't know. Like I told you for PFAPA, we don't know the gene. Similarly, here also we don't know the gene, and but it's No, we know that it's inherited as autosomal recessive inheritance. So these are we diagnose. How will you diagnose this CRMO? CRMO comes with recurrent osteomyelitis. CRMO comes with recurrent osteomyelitis, which is a pyogenic condition again. That's how you diagnose osteomyelitis. Typically, these patients will have skin lesions, acne, psoriasis and all, but no osteomyelitis. These patients will not have osteomyelitis. But that's where Majeed syndrome is all about. Majeed syndrome will have a mixture of features of both CRMO as well as Papa syndrome, where here the patient will have psoriasis also, patient will have recurrent osteomyelitis also. As simple as that. Recurrent osteomyelitis also. Patient will have psoriasis and patient will have recurrent osteomyelitis. In Papa syndrome, you don't have osteomyelitis, and in CRMO, you don't have psoriasis. Whereas Majeed syndrome will have psoriasis, along with that, they will have recurrent osteomyelitis. So this is how you differentiate Majeed syndrome. And then you have other disorders like Blau syndrome and all these things. I told you Blau syndrome may have a lot of features that are similar to Crohn's disease. Like they might have arthritis, they might have uveitis, ocular manifestations, they have skin manifestations like paniculitis, rash and all these things. All these can happen in Crohn's, but in Blau you don't have intestinal features. That's a major thing. So you are not going to have any intestinal features, which is very important. Remember, it's a granulomatous syndrome rather than a pyogenic disorder, your Crohn's and Blau. But DIRA and DITRA are predominantly your pyogenic disorders, where DIRA syndrome will be having more of osteomyelitis, then they have a lot of pustular lesions, then you have periostitis. But no need to remember, these are pus forming lesions. And DITRA syndrome, you are going to have chronic pustular psoriasis. They are going to have chronic pustular psoriasis. So this is what all about DIRA syndrome, chronic pustular psoriasis. So this is what is going to happen. So in DITRA syndrome. And next, what are the other things that you have to know about this autoinflammatory? In a nutshell, in summary, we can put a summary of all these autoinflammatory disorders. Remember, all syndromes will have Mendelian inheritance. They have identified Mendelian inheritance. Every single thing has identified Mendelian inheritance. Exception. What are the two things which doesn't have Mendelian inheritance? One is FAPA. I told you we don't know the gene and we don't know the inheritance also. At the same time, Crohn's disease. They don't have any identified Mendelian inheritance. At the same time, for NOMID also you don't have much identified Mendelian inheritance as well. It's also sporadic, NOMID and SINCA. But these two are very important. Everything, all the autoinflammatory syndromes happens in infancy and childhood. Every autoinflammatory syndrome will happen in infancy and childhood. Two exceptions are the standard exceptions. One is familial Mediterranean fever and second one is Crohn's disease. These are the two things that typically come in adults. They don't come in children, infancy and children. Third one, no fever. Even though fever is a very characteristic feature of all autoinflammatory syndrome, no fever in pustular lesions or pyogenic autoinflammatory syndromes. No fever in pyogenic autoinflammatory syndromes. You don't have any fever at all. And number 4, duration of fever and diagnosis. You can make a duration of fever and diagnosis. Typically, daily fever happens in AOSD and SOGIA. I told you many times. Then you have less than or equal to 1 day. Fever within a day means that usually goes towards FCAS, familial cold autoinflammatory syndrome. Then 1 to 3 days in case of familial Mediterranean fever and Muckle-Wells syndrome, and 3 to 7 days in case of hyper IgD syndrome and FAPA syndrome. Hyper IgD syndrome and FAPA syndrome, 3 to 7 days of fever. Then maximal CNS disease, neurological involvement. Only one, autoinflammatory syndrome with maximum CNS involvement, that is NOMID and SINCA. Very important. This point I stressed multiple times, NOMID and SINCA. And amyloidosis risk. The moment I talk about amyloidosis, you have to think about familial Mediterranean fever. That's it. And finally, last but not the least, all the autoinflammatory syndromes can be treated with IL-1 beta, IL-1 antagonist. IL-1 antagonist will work beautifully for most of the autoinflammatory syndrome. In fact, IL-1 antagonist can be even used as a empirical treatment. If your symptoms and the entire disease spectrum resolves on treatment with IL-1 beta antagonist, that's it. It confirms that you're dealing with the autoinflammatory syndrome. Because all these autoinflammatory syndromes, I didn't tell you that is due to IL-1 beta. This is the cytokine for that is acting as a core cytokine and pathophysiology for autoinflammatory syndrome. So any single disease will be having response to IL-1 beta antagonist or IL-1 antagonist. What are the IL-1 antagonists? You have something called etanercept. You have rilonacept, and you have anakinra. Sorry for that. Etanercept works, but it's TNF alpha antagonist. Sorry for that. Etanercept is a TNF alpha antagonist, but still it works. But these are typical IL-1 antagonists. These two are typical IL-1 antagonists. This anakinra and rilonacept, definite IL-1 antagonists, because etanercept usually works for one thing, that is for TRAPS. That's for TRAPS, because TRAPS is for TNF receptor 1 defect. That's a TNF receptor alpha problem. So that's why etanercept is a TNF receptor antagonist that works for TRAPS. But every single other autoinflammatory syndrome works well with IL-1 antagonist, that is rilonacept and anakinra. Other treatments which you can use is steroids, which standard any thing will respond to steroids. Other thing which you can use is colchicine. Colchicine they will give prominently for familial Mediterranean fever because it's a traditional treatment that works very nicely with colchicine. Third thing you can go is for tonsillectomy, which works very nicely for, I told you, FAPA syndrome, because it's a very important question. Tonsillectomy is one of the very important treatment for which autoinflammatory syndrome you have to go for PFAPA. At the same time, maybe one more new IL-1 receptor antagonist is there, that is canakinumab. Currently it's used mostly in gout, but this is also coming as a treatment option for autoinflammatory syndromes. Canakinumab, this is also a IL-1 receptor antagonist. Typically it's used for the cryopyrinopathies. That's where it's been researched right now. The cryopyrinopathies, FCAS, NOMID and all these things. But anyway, so typically if you ask me in the bracket where canakinumab is mostly researched for, it is for cryopyrinopathies. This is also an IL-1 beta antagonist, cryopyrinopathies. That's where this canakinumab is most useful as per the studies. So treatment wise, IL-1 is the core pathophysiology here. So IL-1 antagonism, by logic, it should clear all the autoinflammatory diseases. So the three IL-1 receptor antagonists, that is anakinra, rilonacept and canakinumab. Canakinumab is especially tried for cryopyrinopathies. And TNF alpha receptor antagonists like etanercept can be used for your TRAPS syndrome. And steroids are a general treatment for most of the things. You can use steroids, colchicine for familial Mediterranean fever, and tonsillectomy especially for PFAPA syndrome. So these are all things that you have to know in clarity, in detail about the autoinflammatory syndrome. I told a lot of information, but the initial first 10 minutes which I have classified, how they classified, that's what is most important. And final nutshell which I have told you, this summary, is what is going to make a lot of sense for your exams, because these are the things that are real exam questions. That's what they're going to ask you. If not, they're going to ask some questions on autoinflammatory syndromes. And with this, we are completing a very tough topic. Hopefully it was interesting. It is interesting for me. Hopefully it was interesting for you as well. So with this, we are completing the innate immunity mechanisms that is due to cellular function. Then in the next section, we might be dealing with the humoral mechanisms and their probable related diseases, which is something very traditional, and you know those things already. So we are going to discuss the humoral mechanisms of the innate immunity in the next section.